Next speaker is Scottie Robinson, who's already had mentioned as the person that Patricia came to work with in Cambridge. Scottie came from Harvard to do a PhD with Barbara Pierce. I think a postdoc with Barbara Pierce, sorry, yeah, in the early 80s. She's worked in Cambridge ever since. Most of that time, I think, at the CIMR, stone's throw from LMB, as I said earlier. And Scottie is a fellow of the Royal Society and a fellow of the of EMBO.
Yes,
just to establish credentials.
Of course. Thank you, Jean. Thanks a lot. No, as Jean said, unlike Daniela, who moved all the way to Singapore, I literally moved next door when I left the LMB. The CIMR where I currently work is right next door to the old LMB. It's a little bit more of a trek from here, but it still means I can come back frequently for talks and things, which is fantastic. Yes, I was a postdoc with Barbara Pierce from 1982 until 1989. And I think like a lot of people, when they find they're going to have the opportunity to work here, on the one hand, I was absolutely thrilled, but on the other hand, I was terrified to be coming to this, amazing research institute with all these famous Nobel laureates. I really worried about whether or not I would fit in particular, because I never really thought of myself as a molecular biologist. I've always been a cell biologist. And I remember a couple of weeks before I moved to Cambridge, one of my friends introduced me to a guy that she knew who had worked at the Strangeways lab, which is a bit down the road from here. And when I told him I was a cell biologist and I was planning to do a postdoc at the LMB, he laughed at me and he said, "Don't you know that at the LMB, they eat cell biologists for breakfast?" So this did not help my nerves at all. But after working here for a bit, it was a wonderful environment and I did start to feel I fit in. I think in particular because, oh no, is this going to advance? There we go. I think in particular because I think cell biology had a bit of a bad name as being such a descriptive science, but most of us really were trying to understand mechanisms. And that, of course, is what the LMB is particularly famous for. So this is one of my own EM images from way back when I was a student. Maybe I need to point this here. Maybe I just need to use the keys. So this is around about 1978 or so. And it was while I was a student that I got very interested in what was a paradox at the time called the sorting problem. In other words, we know that eukaryotic cells can generate and maintain all these different membrane compartments, endoplasmic reticulum, Golgi apparatus, endosomes, lysosomes, and so on. And we knew that there was trafficking back and forth between the compartments. So the question was, how can the cell do this without everything getting completely mixed up? And it was the year that I started my PhD, 1976, that Brown and Goldstein showed that these coated vesicles that bud from the plasma membrane are able to concentrate receptors for LDL. So this was the first indication that the cell could make a vesicle from a membrane, and instead of just packaging a random assortment of all the proteins, the cell could pick and choose. And you can see very similar looking structures elsewhere in the cell, in particular near the Golgi. There's one there, if I can get the pointer to work. There. So this suggested that perhaps coated vesicles could be the key to understanding the sorting problem. But I did realize that you could not really get at these questions by just doing microscopy, which is what I'd done as a PhD student. So I was very keen to work with Barbara Pierce, who in 1975, when she was a postdoc at the LMB, had purified coated vesicles for the first time and had discovered clathrin. So there's her gel of purified coated vesicles with, maybe I need to press harder. Anyway, clathrin is on the far right, the very prominent high molecular weight band. But what I was even more interested in, and this is what I wanted to do as a postdoc, was to find out whether there are other components of the coat, probably not clathrin, that could bind to membrane proteins, things like the LDL receptor, so that they could be specifically packaged as cargo into the vesicles. And so Barbara and I put our money on the 100 kilodalton proteins, which are, I don't think this is going to work at all, just below the prominent band below the clathrin. And together we managed to purify two distinct protein complexes, which we now call the adapter protein complexes, AP1 and AP2. I think my most exciting moment as a postdoc was when I finally managed to make some antibodies against AP1 and AP2, and could show by immunofluorescence that they localized to distinct membranes in the cell, There's AP1 in red on intracellular membranes and AP2 at the plasma membrane. In other words, AP2 is in one place, AP1 in the other place. Now, like a lot of findings, this raised at least as many questions as it answered. We really wanted to know if the adaptor protein complexes do sort cargo, and if so, how. And I knew that this was now a major focus of Barbara's lab, but I had a couple of related questions I really wanted to pursue. Specifically, how do they get to the right membrane? Why does AP2 go to the plasma membrane and AP1 to these membranes near the Golgi? And also, could this be just the tip of the iceberg? Maybe there are a lot of additional AP complexes in cells involved in other pathways. So Barbara, in the spirit of the LMB, was more than happy for me to take these questions with me and use them to start up my own lab. The problem was finding a job, and that did take quite a while. Because normally, having come from the States to do a postdoc, I would have gone back to the States to look for a faculty position. But by this time, I was married to John Kilmartin, who at the time was a junior PI, is now an emeritus scientist. And although he was more than willing to look for jobs in the States, it wasn't a good time. He just switched fields. And I really did not want to go back to the States. I'd become very disillusioned, I'm afraid, as a PhD student, because several quite senior people I knew would talk about their work, and they would hype it so much that, if you knew it that they had actually done, it was actually telling lies, frankly. And when I kind of said something about this, I was told that I'd better suck it up. This is what you have to do to be a successful scientist. And it was so not the way at the LMB. And I felt in the UK in general that I really wanted to stay in the UK. So I applied for lectureships, specifically in Cambridge, without much success. And I think part of the problem was my interests didn't really align with those of any of the university departments, with one exception, and that was a tiny, tiny department, clinical biochemistry, which is kind of ironic because I never felt like a biochemist, and certainly not clinical. And they only had 3 faculty positions, and they were all filled with people who weren't going to leave. However, one of the faculty positions was held by Paul Lucio, who had very similar interests to mine. He's worked on biogenesis of lysosomes for pretty much his whole life. And in addition, they had several, in addition to the 3 actual faculty members, they had several people who had come with their own funding. And Paul was very enthusiastic about my applying for a fellowship and joining the department as a new PI. So I applied to the Wellcome Trust, which had recently introduced their senior fellowship scheme. So that's my actual application, 1988. Project is called Coded Vesicle Adapters to work in clinical biochemistry, with Paul as my sponsor. And to my great relief, this was funded. But as time went on, and I was proposed starting date was May 1989. Just as I was apprehensive about joining the LMB, I was probably even more apprehensive about leaving the LMB, because I had the most fantastic time at the LMB. And I was going to be going to this tiny department, and would I really be able to do the work I wanted to do? So the day I left, I got a wonderful going away present. So this was the students in room 110, which is where both John and Barbara had their labs. So Liz and Ross were PhD students with Barbara, and Mike was John's PhD student. And I should say they've all 3 of them gone on to very distinguished professorships in their respective universities. But you can see underneath there's my piece of film. This shows how long ago it was that we used film. And inside, my blue tips, my yellow tips, my grant supplement. They gave me a pencil and a Pasteur pipette. But I think my favorite is that tube that you can see on the left. So this is Scottie's chemical. Could be anything, better taste it. And this referred to a very bad habit of mine, which would be incredibly frowned upon by health and safety. So of course, I mean, who wouldn't? I did taste it. And I said to the students the next day, I said, that was sucrose in there, right? And they said, you didn't taste it, did you? I said, well, of course I did. And they said, we're going to put sodium azide in there or something. So I said, I did trust them not to. So it was sucrose. Anyway, as it happened, I was able to get my work done pretty effectively. What I was not prepared for, though, was what a culture shock it was, having been at the LMB, because, it was quite an old-fashioned department. And I think Paul Lucio would be the first to say it was rather sleepy at the time. For instance, most people only worked from 9 to 5, and talking about science was kind of frowned upon, so just totally different from the LMB. But I kind of expected this in a way. And I was sort of looking forward, having been such a tiny fish in such a big pond at the LMB, I thought, well, I'm going to a smaller pond, I will be seen as a bigger fish. And people will look at me and they'll say, oh, she comes from the LMB, she must really know what's what. But in fact, that didn't happen because all the PIs in the department were men. And it was kind of as though, by definition, a PI is a man. And of course, at the LMB, I'd worked in Barbara's lab. There are other female PIs, but they just didn't get it. They thought I was Paul's postdoc. And Paul and I would say, no, I'm not Paul's postdoc. And then, they'd introduce me to somebody else. Have you met Paul's new postdoc? So it was quite frustrating. And this is my advice. I decided not to let it get to be too much. But as Danielle has said, you have your passion and you just do your best work. And that's what I tried to do. And of course, in the end, it usually does pay off. There are a couple of other positive developments. First of all, some other cell biologists joined clinical biochemistry with fellowships, including a couple of women. 10 years later, we all moved to what is now called, at least all of us cell biologists working on membrane traffic, moved to the CIMR. So I was delighted that the first director of the CIMR was a woman, Jenny Blackwell. The second director was Paul, which was also great. And the third director was Gillian Griffiths, who many people here know, I think, because she was a PhD student with Cesar Milstein, and she'll be talking this afternoon. So then after Gillian, we had a bit of a hiatus when we tried to find a long-term next director. So Paul stepped in as interim head, but our current director is Julian Rayner, who again trained at the LMB. He was a PhD student with Hugh Pelham. Now, Paul, as I said, Paul would agree with me that the old clinical biochemistry department was a bit sleepy, and he felt this was epitomized by the way we used a bell. So I think everybody noticed, and everybody knows that when we were summoned to come to these talks, a bell rang, and I saw that in the lobby, the entrance, there's this famous bell that was purchased by Francis Crick. Now, the old clinical biochemistry department also had a bell, but it was such an old-fashioned department that we had a tea lady. And at 11 o'clock every morning, the tea lady would make coffee and ring the bell. And at 4 o'clock every noon, she would make tea and ring the bell. And everybody was expected to drop what they were doing and partake of coffee or tea and chat, but never, never, never about science. So Paul said, if that doesn't say it all, that, what they use the bell for. So when Paul took over as director of the CIMR 20 years ago now, one of the first things he did was he went out and he bought a bell, and every seminar he would go up and down the stairs and ring the bell to summon people to come. But in any case, it's worked out very well for me. I think I'm very lucky to be working at the CIMR. And I'm also very lucky to have been supported by the Wellcome Trust on fellowships for 30 years, right up until the age of 67, when I became officially emeritus. So I still have a grant from the Wellcome Trust as an emeritus scientist. So I don't get a salary anymore, but this is my most recent one. And notice the title, Coated Vesicle Adapters, which is exactly the same as my title 30 years earlier. So if anybody had told me in 1988, I'd be still writing a grant application with the same title successfully, I would have been really surprised. But it is just, the field has just opened up in all kinds of surprising ways. So for instance, when I started my lab, we knew about AP1 and AP2. I felt I couldn't give a talk at the LMB without showing a structure. So this is my colleague, David Owen, who like me was a postdoc at the LMB and is now a PI at CIMR. And it was his lab that solved the structure, which has been fantastic. And he's been a great colleague and collaborator. So there it is diagrammatically. And AP1 is closely related with a similar structure that was solved subsequently. And we and others cloned and sequenced the various subunits. This is work actually I started as a postdoc with Barbara. So one of the things I wanted to do was to see if there are others out there, and we used what we would now call bioinformatics, although at the time we actually called it armchair cloning, to find if there are any other AP complexes. And so we found a third, a fourth, and eventually a fifth. This one is actually quite divergent. Now, in the case of AP1 and AP2, they're absolutely essential for life in animals. But interestingly, even though they're all ancient and found in all branches of eukaryotes, as an animal, you can get by without AP3 or without AP4 or without AP5. But that doesn't mean you're healthy. In humans, mutations cause very severe genetic disorders. And this has helped us to understand their function. So I think, in my lab, finding the new complexes has been the easier part. Finding out what they do has been a lot more challenging, but we've been able to take advantage of some naturally occurring mutants, patient fibroblasts, as well as Drosophila at one point. And then when the technology became available, we could do knockouts and knockdowns, and we had to develop some new assays. In particular, we've been able to use proteomics to sort of have a global view of the cell and see what membrane proteins might be getting mislocalized if you don't have a particular AP complex. Oddly enough, the one that has been really a tough nut to crack is AP1 in red, one of the first ones that we found. And this seems to be because if you get rid of it gradually, as in a knockdown or a knockout, cells can compensate. So this is why we, about 10 years ago now, we developed a new method that we call knock sideways, which basically, without going into the details, by adding a drug, you can rapidly reroute your protein of interest onto mitochondria. So this can happen within minutes, and then it's sort of out of action and it can't do its job. And this worked very well. We got some very robust phenotype in the case of AP1, and several other labs have adopted this technique, and it's worked well for them as well. Having said that, we still haven't answered the question about directionality. So that's our current focus mainly, is looking directly at the AP1 vesicles to have a better idea of what they're actually doing. But again, we're developing methods that we hope could be applicable to other trafficking machinery as well. And I've now downsized my lab. So it's just one postdoc and myself. And this means I've kind of come full circle, and I'm spending full time at the bench again, which is fantastic. Okay, just a tiny bit more about CIMR, because I know I'm running on a little bit. So in some ways, it's rather similar to the LMB, similar research culture in many ways, I think, but more clinicians. About 40% of the PIs, of whom we are 25, are clinician scientists. But interestingly, 6 of the PIs actually trained at the LMB. Fulma with Murray Stewart, Gillian, David, and Julian, I've already mentioned, and there's me. And then Simeon was a postdoc with Hugh Pelham. And we also have a couple of second-generation LMB trainees. Susanna was a postdoc with David, and Matthew Seaman was a student in my lab. And I think all of us have imbibed the LMB philosophy. So, the idea that as a PI, you should try to do your own bench work if you can. It's not kind of shameful to be seen with a pipette. Having a relatively small lab and trying to address really important questions, but addressing them not by throwing a lot of money and people, but trying to think outside of the box, which is what I think the LMB is particularly famous for. So I think one of the most important lessons I learned at the LMB was what to do if you're fortunate enough to get a really talented PhD student, one who you could see running their own lab one day. And I think it's important not to be too heavy-handed, not to tell them what experiments to do, but to let them find their own way, give them plenty of freedom. They might make more mistakes then, but it will serve them really well at the end. And, when we all get to the emeritus stage, we look back on what we've done and think about what we're proud of. And I think what I'm proudest of is the PhD students I've trained. And I should say I've always had a small lab of no more than 5 or 6 people, but I've graduated 19 PhDs, and 6 of them are now running their own labs, or at least are about to. So Matthew is in Cambridge. Fiona moved to Australia. She's an associate professor. Andrew is at University of Sheffield. Alex moved to the States, Georgetown University. And my most recent PhD student, Alex Davies, is currently a postdoc in Germany, but she has a couple of very nice offers to take up a PI position back in the UK. And of course, the other student I'm incredibly proud of is our former speaker, Patricia Kozik. So, for both giving me a direction of research and a philosophy of research, I'm just incredibly grateful to the LMB. So thank you, LMB.
Richard.
So, do you know, can you prove that there is no AP6 and AP7?
That's a good question. No, because AP5 was so divergent, but there are some tools available. We did find one that we call TSET, which is more like COP1. We developed our own program using, HH-PRED, which kind of, long story short. So we were able to find another one, which is lost from animals. But, it's when it's just the structure that's conserved, it could be related and just impossible to find at the moment. So there could be. All right.
Marianne.
Plants have this system.
Yeah, plants are great. Plants have got all 5 of the AP complexes, as well as TSET, the one that we've lost. And they're fascinating. I went to a plant meeting as the honorary cell biologist back in August. And there's some really nice genetic studies, which I think because AP4 and AP5, we don't have that much of them. Plants have a lot more. And I think they provide insights into what's happening in animal cells as well. I think looking at the whole diversity of eukaryotes is really important for this kind of thing.